statement from Andrea. Now, back to the point. How accurate are pairwise alignments? So we're back in the lecture. Oh, now we get online, right?
Yeah.
The whole time?
No, but just when we start.
Anyway, so far in this lecture what we talked about is simple pairwise alignments. Dynamic programming, hashed, BLAST. How does it work? How good are they? Well, let's just begin with a few simple statements. Does a similar sequence imply a similar function? Yes or no? We have both reactions in the room. Sometimes. I believe the no reaction has changed to sometimes. Does a similar sequence imply a similar structure? Do I get the same reaction? Sometimes yes, sometimes no. Everybody agrees to that. Is there any differential between the two? One of these two is more true than the other one? Yes, but now one of them is more true than the other one. I believe that's what I deduced from your head shake. But which direction?
I will say function.
Function is, so the statement is more true here than here, is what you say. Okay, let's get at something else. So what we look at here in structure space and sequence space. Structure space, again, is smaller than sequence space because there are more sequences than structures. Whether you can condense it onto the same thing is exactly what we want to look at, and soon we will get the answer to that. But let's just look at a few realities. So the first reality is that two proteins relatively close in structure space, they come from different points in sequence space. Okay? That's a very clear statement. And again, so essentially you all agreed that something like that here is true. If you're close in sequence space, you will end up close in structure space in many cases. So there's exceptions. Now, what about this one here? Most sequences do not fold at all. Is that true or not? True or false?
If you consider random sequences, then yes. But in biology?
What do you mean in biology? So biology does not have access to random sequences, is what you're implicitly saying?
The sequences that are expressed are most of the time found.
So we are getting into, you're saying too many things at the same time, and if you keep going then you will say everything and that will explain the universe. But let's begin at one point. So the first statement is that I can imagine protein sequences. Do you believe we can imagine protein sequences that we have not observed and that will fold? Let's begin there. Yes. What is the evidence for that? Have you heard about any evidence? I briefly mentioned it in passing, but I completely understand that you didn't catch that. It was one of, I speak for 90 minutes, and that was a tiny, tiny statement in 90 minutes. So one evidence is that the more, when we scoop up from the soil, for instance, new bacteria, then we scoop up sequences that we have not seen before, constantly. And those would, in fact, is a subset of sequences that we could imagine, haven't observed yet, and suddenly we observe them. Okay? So there's a lot we have not observed. That's the one important statement. The other important statement is very much along the lines of what you say. Most likely, in fact, most sequences, now you said two things. One is they cannot be expressed. So we don't quite believe that. So it is true. We cannot, in a computer you can change every residue against all the other 19 amino acids. In a single nucleotide change you cannot due to the genetic code. But still, you have an immense amount of possibilities in the genetic code through single mutations. And most likely, most of those, and that was your second statement, will not fold. There will be something wrong with them. So most likely, most sequences that could in fact happen, and that do happen through mutations, will not survive. Most extreme damages are so bad that we don't observe them. because they don't exist. And that is an important aspect here. So the space of all possible sequences is much larger than the space of sequences we will ever observe. Although the space of sequences we do observe today is much smaller than the space we will know in 10 years from now. So we are far not close to saturation as we had believed 10 or 15 years ago. It's not true. We add every year as much novelty as we did last year. No matter how much we add, okay? The amount of novelty is seemingly not changing. That, in fact, in some sense, is a total surprise. Had absolutely not been predicted 10 years ago. And in some sense, you could say it's a riddle. But it is true that the sequence space is clearly much larger than what we appreciate today. And it's also true that most of the things most likely will not exist. Now, back to that issue of the zoning here. We have sequence identity. So our plot here is the identity of residues between two proteins, 100% sequence identity and further down. There is a zone that you may call a daylight zone. That's simply the zone where two proteins are so similar that by simple pairwise alignment you can recognize similarity in structure and function. Okay? There's some zone there. And we are going to get back into where exactly that zone sits. Then comes a zone that has been referred to as the twilight zone by, in fact, Rost. And the twilight zone means that, so here you have the light. You can recognize the similarity. From sequence similarity, you infer similarity in structure or function. Here, you somehow can. You still see that something that is similar, but it could be wrong. That's why this is the twilight zone. That's the midnight zone here. In the midnight zone, there's just no signal anymore. Meaning that most of the similarities from sequence alone that you find in that realm are, in fact, to things that are not related in sequence or structure. Okay. Now, what I show here is one possible. So what we now want to know is when we take pairwise alignments, how is the transition from that, where we in fact have no problem of doing pairwise alignments and finding the similarities, to the region where we can actually not do it at all. How's that transition going to be? So one way in which we could imagine this, we could guess this, is if we looked at the percentage accuracy here, for instance, then 35% sequence identity, so somehow that's where the twilight zone typically sort of in this area is defined. And then the signal drops like that. Right? At the same time, you would somehow see that the true positives would shoot up. You would find more things, but the false positives would shoot up more, right? Somehow something like that is what you expect. How can we assess the alignment accuracy? How can we, in fact, we have an alignment method, BLAST, dynamic programming, Smith-Waterman, or whichever you pick. How can we see whether it's right? What can we measure? What is a good standard of truth for assessing alignment methods? Again, we have methods such as BLAST, you all know it, where we simply compare strings of protein sequences. There's something about this Thursday that makes everybody, the entire room, was there an event last night? Or was the entire room and I also feel not fit, and I know why, but somehow everybody has a tough one. But this is too cold to open the window, I'm afraid. Anyway, so back to the question. How could you imagine to check how well alignment methods work? Do you want to say something, or was that just you move?
I want to say specificity and sensitivity, but I'm not sure.
But how? Yes.
Looking up true positives, true negatives, positives.
Where do you get them from? Exactly.
Testing.
Huh? Yeah, but so again, yes, that's what I'm showing in these plots here. I talk about true positives. Who gives you true positives? Who gives you false positives? Who tells you that a particular alignment is right or wrong?
You have to check against some real structure in some real structure comparison, and if this works on the sequence sequences, then
So what are you trying to say? You're trying to say that you have a pair of proteins for which you know they are similar in 3D, through a 3D comparison. And now you say, my alignment, does it catch those? Is that what you mean?
yes.
Would that work? Would that be fair? Would I compare, so on the one end I do a structure comparison as true and to give me the blue and the red, and on the other end I do sequence comparison to give me essentially what I test. Is that sober? Is that kosher? Am I mixing things? Is that okay to do? Nobody has a problem. So yes, indeed, that is. So we can in fact do, take PDB, do an all against all, and then do the same with sequences. And then we are going to use 3 classes. We are going to say that if there is less than 2 angstrom, 2 proteins are related. We're going to say if there's more than 5, they're not related. And then we're going to say that in between, I don't know, I'm going to throw all the cases out, just to not make mistakes and calling them red or blue. I have a realm, and very often this type of procedure, again, this is something that very often you should do. Too often people, in fact, put the threshold for a decision between red and blue onto the middle of the Gaussian, so that you make maximal mistakes in the assignment. So try to have a zone in between. Anyway, now we can do that. There is a slight problem in here, and that problem, we cannot really do all against all, because that is too much bias. You square the bias in some sense. So you have to take out a sequence unique subset and do that sequence unique subset against everything. That immediately brings up the question, why not do sequence? So if bias is a problem, why not do sequence unique against sequence unique? So I say do an all against all is bad because PDB is biased by too many things that are too similar to each other. So that's why I have to cut out a set that is such. Sequence unique means there's no pair in there that has more than a certain level of sequence identity. And I take that one out, say PDB has 100,000, say the set is 10,000, and do 10,000 against 100,000. Why can I not do 10,000 against 10,000? That would be maximally unbiased, but why can I not do that?
Because then you don't get similar.
Exactly. By definition, this set is such that I find no similarity above a certain threshold. So I cannot check things that are above that threshold. That's why. And then, so that is exactly what Chrysander and Reinhard Schneider did, published in 24 years ago, in the original, what they call the HSSP curve. The HSSP curve looks the following way. So you have number of residues aligned versus percentage sequence identity, and then the curve distinguishes blue, essentially things that have a similar structure are here, red, things that don't have similar structures are here, blue things are also here. So this curve essentially is a curve that distinguishes know from what we cannot know. There's a slight refinement of that curve that comes a little bit later, so that adds a little bit more detail. This is the gray one here, but in principle it's the same thing. Let's look at the data. The actual data underneath, red things that do not have the same 3D structure, blue that do have the same 3D structure. You immediately appreciate that there are tons of blue points under here. So the curve clearly is not such that there is no blue under. It's also not true that there is no red above. But you may or may not agree with me that this seems to be somehow a line that separates out red and blue. Is there, if you looked at the data this way, if that were the data that you produced in your project, is there anything in the way I show the data that is not clear enough, or is there any question that the data asks, or is there anything somehow, when you see the data, there's something wrong here. There's something that you should jump at. Is there? What is it? I don't know how to answer my question without.
I have no idea how Many points now.
Exactly. That's the major point here. So what these images suggest to you is a density. Because you have many points. That gives the brain the idea, since you have many points, that in fact what you see is every single point. That gives you the idea that you actually see the density. But it's not true, or it may not be true. It may be right, or it may not be right. And in fact, I'll show you numbers in a moment here that will show you that this is visually completely misleading. So there's actually very little red above here. And the degree to which you have blue versus red, in the density here, you may even say that there is not much more blue than red. With just the red clusters here, and that is not the case at all. I'll show you in a minute. Yeah?
Sorry.
No. If it is for the lecture, it's fine. Otherwise, you don't have to be sorry. It's just... so there are a few points that I want to make. The first, there's a saturation point. So there is, in fact, what level of sequence identity is important in pairwise alignments depends on the length of the alignment. But that is true only until 300 residues or something like that, or 200 something. Then the line goes into saturation. And there's another extreme line here. So if you have 11 identical residues, you cannot tell whether they have similar structure. Why is that?
Could be just alpha helix.
Yes? Alpha helix is different than a beta strand.
Yeah, but you don't know how it's aligned in the structure.
No, but if it's an alpha helix, so if those 11 residues are always an alpha helix, and another 11 residues are always a beta strand, that's the difference. And you could say yes. You can immediately say blue and red, right? Helix, strand. If you match a helix on a helix, this is more similar than a helix on a strand. So you could say something about that. So ultimately, the point here really is that when you're too short, you have long range constraints that are missing in order to fix whether that is a helix. So there are 11 mirrors that at one point in a protein become a beta strand, and in another one in a helix. The same thing, and I will show you an example for that later in the lecture. Just an example here in this particular structure, and I can't remember what the structure is. I have 150 residues in this alignment at 35% identity. That is above the line. While in this particular example here, in both of these alignments are in fact those that are functionally important. The example from Marco Punta. Here we have 40%, which is much more than 35, over 50, but that is below the curve. So it's not a question of 40 versus 35, but it really is alignment length. So the density is the problem that we have to look at. Now, what we're going to do in order to learn about the density is we're going to put slices of this curve. We're going to look at the distance from this curve. So where the curve is here, that's the distance +5, +10, +15, and so forth, and that's a minus distance. Below the curve, above the curve, right? And now I'm going to look at each of these slices, and I'm going to ask how many red and how many blue are above that curve. Okay? That's what you have here. That's logarithmic scale. That's this distance from HSP threshold. This is the corresponding sequence identity alignment for the point at 100 residues. So the actual thing that I'm showing you is the distance to the curve here. Okay, you see that we come at some point here, then we go into the twilight zone, and we increase the things that we could find by more than an order of magnitude. Right? So this immediately tells you why you want to intrude into the twilight zone. Because here, for this experiment that I did, whatever the numbers here mean, essentially things I find, but the point is, this is more than 30 times more. So going from 35 to something like 15%, or from 30 to 15, going 15 percentage points further down. So I go from 100 to 30, and then 15% more, and it buys me a factor of 20 in the things I could find. I find 20 times more things than I found from 100 to 30, right? That's amazing. And that immediately tells you why you want to get in there. Okay? But now comes the bad story. That's the wrong ones. So at a point that is not far away from 30, you're essentially at 0. No mistake, that means. There's some mistake here. There's a big mistake here. I don't find this problem. But anyway, so the point that I want to make here, what is important here, is that I go from, say, 30% sequence identity to 15% sequence identity, and in my false positives, I move by 4 orders of magnitude. We're no longer talking 20-fold. We are now talking 10,000 times more. So at the same time that I win 20 times more right ones, I hit against 10,000 times wrong ones. And that is an amazing battle to win, right? And that immediately tells you that the transition here, that the transition is a rapid one. In physics you call that a phase transition. So there is really a big step that happens somewhere here in the twilight zone that is defining the twilight zone. And in fact the reason why this happens, there are 2 reasons for that. One is mere statistics, and one has to do with sequences or with the way proteins make contacts. But statistics in principle already explains why there is a transition that is a phase type of transition. It's a cooperative effect. So the way you can think about it is you have a mutation that may be damaging, but then you have another mutation that you can cover to compensate for the previous one. And you can do that one at a time. And at some point you get to the point where you can no longer do it, and suddenly the whole system falls. Suddenly you can no longer find, for the next mutation, you can no longer find any substitution because you had already too many before, and then boom, with one fall it goes down. That's the reason why it's a cooperative phenomenon, because proteins, whether it works or not, is something that is defined through the network of contacts, and that is intrinsically a squared entity. And that explains why this is not a linear phenomenon. Okay, back to the first question here. What more could you do? And the simplest way is what at some point was called sequence space hopping. So with a pairwise, what this curve gives you is you start from a protein A, and you essentially define a circle around it. Anything that is a certain level of sequence identity with respect to the alignment length, with respect to my protein, I say yes or no. Okay? Above the curve, yes. Below the curve, no. That defines a circle. And the same is for these 3 proteins. Now assume the 3 proteins A, B, C are such that in fact their distance is too high for the safe zone. So you can in fact not tell whether C and A are related. Too far away. Assume that now you've found a protein X which is in the safe zone of A and is in the safe zone of B. Since A and B are the center, they are not, this distance is too high, but this distance is okay, this distance is okay. Now what would that imply? So you can clearly say X has the same structure as A. And you can clearly say X has the same structure as B. What does that tell you about X and B and A? They have the same, provided we don't have the domain problem again, right? So we have to make sure that we're not running into the domain problem where we have something like this here, X, B, A. Where in fact in this particular case here X could be related to A and X could be related to B, and B and A are still not related. So as long as we exclude that, it appears that this could help us to infer that A and B are similar. And that is what this one shows here. We remain at 100% accuracy deep down into the twilight zone when we make these hopping inferences. So remember, this is where previously we had drop to much lower levels here. At 0 we dropped like this. So we're down to -10 here, and we're still staying up here. So essentially, instead of going down here, we stay up, as long as we can do that. But what else could we do? So that's what was a hack if all you ever could do is pairwise alignments. What else can we do? Better than pairwise alignments. Yeah. Sure. So multiple sequence alignments, again, I showed you how we do dynamic programming on 2 sequences. This is the optimal solution. Multiple alignment simply is the same thing. In this particular case for 3, we would have a 3D matrix. In principle, that would work. And in principle, again, you can show that you find the solution, except for now we hit an NP-complete problem. NP-complete, essentially, you may have heard that, means this is not solvable in linear time. We can, in fact, do it these days up to 6, but then you need 60 terabyte of main memory. I'm not sure whether that's true. I read that somewhere. But anyway, we see that very early on, with 6 we get into severe problems. Do you have any idea how big families are? Is 6 a big family or a small family? Small. So whatever the big problem with 6 is, we're actually targeting something bigger than 6. And that is clearly beyond the reach. So for more than for more than 3, 4, 6, we cannot easily do that. How could we do it? So the next slide already proposes the hack. Can you think about a hack? So the first hack came from Lippmann a while ago, and that is a pairwise dynamic programming in the vicinity of the intersection searched n-wise. So essentially you're cutting out small bits and you look at small bits in different ones. That speeds it up is one hack. Here is the hack as old, and that in fact is the hack that we still use today. And that comes from Rost, the man with the Twilight Zone word. And the idea really is map it to a tree. Map the problem to a tree and then do pairwise alignments. So here's what I mean. You have to align A, B, C, D, and you simply begin by all pairwise alignments. You build up a matrix, and one way is you begin with a first pair A and B. So now you align A and B. Okay? It'll come up with one way of doing it is with a consensus sequence. You're familiar with the term consensus sequence? Okay, in step 2, you can now have 2 different ways of doing that. One way of doing it is you can take the next most similar pair. Here, these are the green and black here, and come up with the consensus sequence. And then in the 3rd step, you align the consensus sequences. And you have essentially done something that has a feature aspects of a multiple alignment without ever leaving the space of doing pairwise alignments. Right? Can you see any other way in which you could have done the same hack or a similar hack?
Aligning the consensus sequence to...
Yes. So you could go down the stack here. You could take that consensus sequence, go against the green with the consensus sequence, and then go further down and begin building consensus sequences that are aligned. And you could have chosen this idea here, and you could have chosen the path in which you separate them in a different way. Again, so we want to intrude into that zone here. I'm not sure what I tried to explain with this slide. It's the last slide that's hanging. Okay, so that is one hack. Let me take this question or general statement or... So the next level up from here is to use, in fact, the multiple alignments and build profiles. PSSMs. What is the difference between a profile and a motif? Do you all know what a profile is? You don't need to, because I'm going to show you some slides what a profile is. Okay, let's begin with the following thing. We have again this pairwise multiple alignment built up here. We begin to build the alignment. We get something like this. We build a family alignment, and this family alignment here, again, what I mean here is that is a sequence aligned. Here is some significant similarity to different sequences. And now these slides come from Teresa Word. We can take essentially, sorry for the animation, we can take here what we observe at every single position, and this particular is a gene alignment for transcription factor binding site, but it's the same idea, where you take every single position and you count how many, or what is the information content of the particular residue that you observe at that position throughout the family. Okay? Where in this particular, I don't know whether you have seen the letter plots, the height of the letter is proportional to the information gain from that letter. And a motif is a slightly different story. Motif is typically written like this, so you have 2 or more possible amino acid, leucine or isoleucine here. You have some that are disallowed, so you're not supposed to have a positively charged arginine or lysine there. You have some repetition from 1 to 3. You have any residue, and those are 3 sequences that would match with this motif, but many others would. That's different in the sense of a motif. A motif really is in the sense a regular expression, while a profile could be carved into a motif, but typically has not this regular expression, and it's not that many motifs map to it. A motif typically is something that has a probability. Sorry, a profile, you have a probability to map to a profile, you have a yes or no to map to a motif. That in some sense is the difference, and then unfortunately there are technology, or there are means to sort of blur the borderline and to find ways between this. Okay, let's get back to the substitution matrix. A substitution matrix, again, is simply the blossom, for instance. I compile how often do I see a certain substitution, how surprising is it to do an A against a C or something like that. Now, building a PSSM, you begin in fact exactly with this. Well, the first level is you begin with pairs, then you put in the substitution matrix of blossom. That gives you the first frequencies, and then you build up the family. Let's see that in an example for the for gene sequences. You begin with an absolute question. Begin with an absolute frequency, you turn it into relative frequency, you put that into a table. The table leads to log odds. These log odds, that is in fact the corresponding matrix, the blossom matrix type of log odds. And now you in fact here build up this profile. The more sequences you align, the more you replace the blossom matrix against the specific matrix, position-specific substitution matrix. That is essentially the profile for that particular family. PsyBlast. PsyBlast is doing exactly that. So again, the idea is you build this, you use this family. When using this family, you see that there are particular positions that are conserved, and those are more important to align. You begin the first step with the general, and then you turn the general into the specific. The people behind that, so there's the PsyBlast formalism based on BLAST, Webb Miller, so the Stephen Altschul, David Lippman, Tom Madden. Stephen Altschul, I believe, did I present that before? So Stephen Altschul is an example for somebody who has changed the world with relatively few publications. 70 publications for somebody who has 2 that are quoted over 50,000 times. So this is incredible, and well, that's BLAST and PsyBlast. That completely has changed the world. You can, and most of the work he is doing is essentially improving BLAST that is not published. So there are examples where you can just refine a tool if you're successful. Refine a tool, refine it, never write about it, and then every decade or so, or the last publication is more than a decade ago, you get something that changes the world, and that's good enough. David Lippman is somebody who comes from mathematics, 71 publications, but what this one hides is that David Lippmann has published actually a lot of things with very original ideas. And he is part of BLAST, but he's also part of FAST-A, the alternative early method that did it. The concept of PsyBLAST is, as I said before, is the fast hashing. You put things into words that you can quickly match. Given the match, you extend the match in both directions. Here the seed, you extend the seed by dynamic programming. That is much faster because you have the square of the length of the pair, and since you have short words, you're very fast. In between, you don't align. So this is essentially the way the BLAST works. I don't want to go into the detail now. Then that's the dynamic programming extension that I talked about, and as I said, in between you don't know. What you need in order to make PsyBLAST work is the same as for BLAST. You need to work out the statistics, background hits, difference with respect of the mean value here with respect to the standard deviation gives you the e-value. The e-value tells you what to accept and what not. You compile statistics, you collect all pairs, and build a profile. Once you have that profile, you do the I of the PsyBLAST, the iteration. You do it again. So you do again with this profile now against the database, and you refine while you go the position-specific profile. So let's look at the sequence profile, or the sequence space, to explain some strength of this profile-based approach. PsyBLAST is just one. That's valid for all profile-based approaches, and PsyBLAST was not the first. So you begin with a sequence U, and say the green were the family that you want to match, that you want to find. With a simple pairwise alignment, you have access to this blue disk. Because that gives you simply a very symmetric pairwise alignment. Anything you find at a certain distance is symmetric around the middle point where we started. Now, in this example, I assume that the database is such that the hits you find in the safe zone all happen to be on the right side. Okay, the implication of that is clear, right? That means these hits, they in fact will decide how the profile looks. And this means that the profile now, in the next run I go out, will find things in this region here, slightly shifted to the right. The lens, the white lens here is has a larger area than the blue one, because the profile reaches further, but it's no longer centered around the same point, simply because it's driven by what you found, right? One important implication of that is that if you had one particular hit here, in the first iteration of PsyBLAST, this hit is lost. So something that was a very safe hit, if you do something that is better, the position-specific iteration in the second step, and you lose what was good. Is that really happening? Yes, in every single BLAST run, and many. So for some sequences it happens more often than for others. It depends a little bit on the particular sequence you use. But this is a very common phenomenon. And when you run PsyBLAST 5 times, you have to go back. So PsyBLAST does not collect for you. At the end, you get a list. This is not the sum of all the parts. So this is somewhere in the top of the file, and unless you parse the BLAST right, you will not see that in the end. If you only take the best BLAST result, typically, if everything goes fine, is the last one, but it will still not contain things that were right before. It's one thing that you have to remember. And there are good reasons why that is so. Here's another thing that it does not do, and for many years people have done that as a hack. Now it's no longer easy because of CPU time. But so remember I told you we could in fact the sequence space hopping kind of idea. So I could draw a circle around every one of the ones that I found in the first moment in the first safe circle. And I could have used all of those, and those are these light blue disks here, as starting points for new searches. And whatever I found with those is sort of the sequence space hopping idea, and I showed you that I essentially stayed at 100% accuracy doing sequence space hopping, as long as I don't hit the domain problem. And the area of this light blue here is larger than the length of the white. So PsyBlast is not doing sequence space hopping. Again, the reason why it's not doing that is because of CPU. But this was one technique that people used to predict structured CASP, because it reaches further. Here's one of the problems with any approach such as PsyBlast, any profile-based approach, which is that the moment my lens gets too big and I pull in a protein from a different family, then I suddenly pull in a lot of wrong ones. Why is that a sudden event?
Because when you have one of the other family, then in the next iteration, it will get more of the other family and then it starts escalating.
So the reason why it's so sudden is because the further detailed you go, the more you get down to the fact that there are only 4 residues that are conserved. And these 4 residues are the most important thing you should look at. The moment you get one wrong, this entire information is wiped out again. That's why it's so sudden. Because you exactly violate what you have evolved until that point. Now, how could you avoid that? How can you make sure that when you run PsyBlast, so again, there's no automated way in which PsyBlast does it for you. This is up to you. How can you possibly, what could you invent that could help you to see whether this has happened? By the way, when PsyBlast came out, the simple solution was to cut the number of times that you do it. And in the beginning of PsyBlast, we ran PsyBlast 2 or 3 times only, because after that, essentially for every single time there was a pollution. That has changed, because there was a lot done in the background. Now you can easily run automatically PsyBlast to 10 iterations and typically nothing goes wrong. But still, how can you find out whether that happens or not?
Perhaps if the profile after one further iteration changes drastically.
Yes, and that's in fact exactly. So there are 2 ways of saying that it changes drastically. So every single column could change dramatically, or the number of sequences that I pull in. So, you know, the most extreme example, suddenly I pull in the database, or I pull in some significant fraction of the database, and then I know something is wrong. And those were, in fact, tests that we did in the beginning to have some safeguards. The degree of change of a profile, which in some cases it was real. So in some cases you open to a different end of the family, and you suddenly really pull a lot of, as many things as you found until that point. Sometimes you can run 10 iterations, and the 11th is going to bring you more sequences than you had so far. And this is all true. Because you discover a real new relation. And that's exactly what you want to find. But sometimes the factor of 2 indicates that something has gone wrong. It's still not that easy to distinguish these 2, but we have 2 ways in which people sort of do safeguard. Unfortunately, there is no clear solution other than a hack that you as somebody who applies to PsyBlast does. So the developers of HHBlitz or PsyBlast cannot really solve the problem for you. And I mentioned PsyBlast because it's by far the most used method. cont'd is in principle doing something like that, except for PsyBlast does the finding and the alignment. cont'd focuses only on the alignment. Let's now get into Clustl. Clustl. There is a Clustl V, a Clustl W, Clustl X, Clustl Omega. These are the dates. You see there is quite a span of development time. It started with Des Higgins, and these are some of the developers that are on the recent paper. Here's the idea. we begin an all against all. Essentially, I showed you the same slides before, but dynamic programming. So now this means here, the assumption, the starting point is we had run a PsyBlast before. So somebody, whoever runs Clustal, somebody has given to that person a data set of 1,000 proteins that are going to be part of the family. The only game that, or the only question that Clustal addresses, how to best align those 1,000. Okay, so again, you take the 1,000, you do an all against all, build a phylogenetic tree, and use the tree to decide how to in fact align the pairs. So how to group the pairs and then do pairwise alignments. Now, that brings me to the issue of phylogeny. Reproduce phylogeny. Let's assume that the following happened. We had, at some point, we had a gene, and there was a duplication event. Is that a common thing? That you duplicated a gene and you do, some offspring has twice the same gene? Absolutely common, in particular in many bacteria or yeast, or very, very common. Now, we go on and we begin to, since we essentially, in this particular case, what you would typically immediately assume that we have two domains, or twice as many domains in the duplicated version here. So since there is the blue part here that can do the function, we have a light blue part that can evolve away. So that can slightly change. Because the function is already done with this one, we can use this part here for something else in the new organism. And then you can imagine that, now we have this two-domain protein, and that in fact is again duplicated, again a very frequent event. Things change over time, and then over time at some point this completely changes into this red thing. Story 2. We have the green one here that in fact is the other way around. So we have a cleavage event. The cleavage event has a similar story. We in fact have two different sites. Something evolves away. We have a duplication event again. Something evolves away. Story 3 is this one here, where again we have a two-domain protein that sort of at later points in the evolution picks apart. Those are my 3 stories. Each one of those I invented is true by my invention. Now, say that they didn't survive. Say what you actually see from all of these events is this. Then the problem is that you are inclined to align them in a way that looks like this. And that alignment is absolutely not relating to reality, to the history, because you don't have those trees. Most of the organisms that have lived are dead, did not leave any genetic material. Most of the trees are completely incomplete and will never be complete. So what we do in our alignments is a fiction of phylogeny. That's something that we have to always remember. But biologists like trees. Fiction or not, they sound good. And there's certainly one way in which you can organize the data. And for the Clustal, that is enough of a reason to begin with trees. The trees are not trees. One of the, so then you go A and B, then you pull C into it, and then D into it, according to the tree, and you keep going. The number of sequences versus time, it goes up. Different versions of Clustal Omega in red compared to other methods, and you see that they are all dynamic programming methods here. Some are a little bit faster. Clustal W overall is the message here, is not doing too badly, but in fact the real thing is they do very well in terms of the accuracy. But you do see that there is an issue about the explosion of the CPU time that you need for growing families. So today, finding 3,000 in a human family is not unusual anymore. And over the example that I just had, 6 multiple alignment with 6 somewhere down here, and you see there's a tremendous increase in the CPU time. So Clustal W has an issue with time, or Clustal Omega. So essentially what this one here says is that with iterations you can improve accuracy. So with Clustal W you can do iterations and you can do better, and you do better every single time. Over 100,000 citations. In fact, one of these cluster papers, I believe, is the most quoted paper in Science. Altogether, that's Des Higgins here, 94 publications, 2 over 30,000, H-index over 52. Toby Gibson is a group leader at EMBL, over 70,000 publications, 70,000 citations, 128 publications. Now, the third person is Julie Thompson, and so she inherits many of these papers. What is so amazing about this team, I believe, is what explains why, in my view, why cluster X omega is so successful. This is the guy who does the algorithm. This is the guy who's the biologist. Toby is a person who can look at an alignment and essentially say, I believe this or not. He has an amazing feeling for alignment. He does this by coloring his sequences, by the features. That is why cluster W is colorful. That is the program, that's the part that Julie added to that, the output that I believe is very appealing to many of the users. So we have something where the method is right. It's not the best method, it's just the fastest hack. The method is put in a way that experts trust it, and this is done by having the right visual interface. So I believe 3 things come together in Clustal in a unique way, and that ultimately makes Clustal so successful. Everybody can use Clustal and put the result of Clustal easily. Every biologist can put the Clustal figure into their final paper. You cannot do that with BLAST. And I believe this comes from Julie, that part, maybe one of the biggest reasons why Clustal in fact made the day. There are other methods out there. So this clearly is the representative for dynamic programming. I'm mentioning Maxom here, and I'm mentioning Maxom ultimately because of the different philosophy. Reinhard Schneider and Chris Sander, I showed you those 2 in the HSSP curve before, the length versus sequence identity curve. So the big difference in the philosophy, this is all similar to Clustal. It started at the same time somehow. The details are different, but the major difference is the one from protein-centric to family-centric. What I mean here is that in Maxom, the philosophy is that the protein you start with is a protein in which you don't do anything. Don't do anything means no insertion, no deletions. That's the protein for which you want for every single residue to know something. Clustal W, the moment it goes out into the phylogenetic tree and decide which pairs to align next, completely lost. At that moment you completely lose the idea that there's one sequence you're interested in. That view says you're interested in how the family is aligned. That means in particular that for this first sequence you have insertions and deletions. You are working as an experimentalist on one particular protein and you want to know whether that arginine or lysine or whatever that is, which one did I do? The serine here, whether that serine can be mutated. From Clustal you will not really see that because it's just not there, right? That's the downside. Again, there is, if you subscribe entirely to this philosophy of a family, I write Clustal, HMM, and we will get to HMM and T-coffee to the other methods. They all have the same philosophy. And unfortunately, the only method, PsyBlast has also a different philosophy, but PsyBlast has the issue that you can have regions that are unaligned. So PsyBlast has its own issues. But there is, in fact, Maxom was sort of the last of its kind method that clearly pulled through this idea everything is subject to one protein of study. Okay, another way to do the math, hidden Markov models. Do you know at all anything about hidden Markov models? Have you encountered anything like that before? Yes, you have. Okay, so then it's very simple. So we have we have states, we have transition probabilities. There's a transition go back into the state 1, 2, and probability again to have an end that creates these, and the states in fact are hidden. The observed symbol sequences here come out of it. Those are emission probabilities, and you, in fact, for the particular state sequence here, you could imagine different sequences. So you could have different observed symbol sequences that are compatible with this initial one. This is just like a motif. Now we are probabilistic, but in principle that is the motif idea. The slides here come from Marco Punter. So in an example here, Marco likes football. This is from the last season, and I'm not entirely sure why he picked Augsburg. But anyway, he picked Augsburg, and he simply asked for Augsburg playing at home, what's the score in the last season of the German championship from August to November. Those are the results. Away means, wait a minute, what is it? Red, blue, I'm not sure what the black one is. So then here comes the score. It says a loss, win or draw. So you win, lose or draw, and you can in fact imagine a very simple hidden Markov model that does the probabilistic system here, and to model the states of the table that you have seen before is very trivial. And the one simple way in which you can use that as an input is home away. You simply ask, are they playing at home or not? Maybe that's the only input you need to predict the outcome, win, draw, or lose. Okay? Hidden Markov model could easily do something like that. There are probabilistic models, finite state of states, discrete alphabet, same thing. Okay? I'm saying the same thing again. What about this one here? So I can model the same thing, right? I can have states. Is that a hidden Markov model? No, it's not, because this is absolutely deterministic. Here we don't have any probability. There's only one sequence of events that is compatible with the model here. And that is the big difference, right? Now, whether we again could imagine that we have a probabilistic model that we can get from the pressure or something else. Here's another example for the dishonest casino. In the dishonest casino, we have a fair die and a loaded die, so one that doesn't really give you the right values. So it does not have the same probability for all numbers. That's what a loaded die is defined. And the probability at which the casino switches between the two die is a certain probability, so that nobody can easily see that. So once in a while the loaded die comes with a certain probability of going into it. Let's just have every fourth time the loaded comes, and once I have loaded more, at least some half of the time or something, I go back to the fair. Right? For users or for onlookers, this is very difficult to appreciate that there is a loaded die. Just if you see what the numbers of the sum of the two are, you will not immediately cry that something is wrong, right? If you had that alone, you would most likely, after 5 throws, you would sort of suspect that something is wrong. But with this system, you will not. You can easily model that with a hidden Markov model. That is exactly the kind of thing it does. Okay. Into alignment, so there are the two major schools that do it, SAM and HMM, or Hammer, where Hammer comes from Sean Eddy, SAM comes from Kevin Karplos and David Hausler. That always started, and I put this name up, Anash Koch. Anash started working on neural networks, and then wanted to, Anash, as you may infer from the name, is from Northern Europe, is Danish. And Anash had this idea that Copenhagen is cold, and there are places in the world that are warmer, and when I do a postdoc, I want to do it somewhere where it's warmer. And he had the idea that Santa Cruz is a nice and warm place at the Californian coast, and bumped into David Hausler, and David Hausler is sort of a Hawaiian shirt guy who's always open to any idea. So he had this idea of hidden Markov models for alignments. When he did that, so David, as a physicist, was interested in it because he just liked the model. He was a theoretical physicist. He liked the idea of hidden Markov models. But nobody at the time that Anash went there in '92 had ever done that. And nobody had any reason to assume that we need anything other than BLAST or than dynamic programming. And there was this relatively young guy who completely changed the world. well, together with David Hausler and Kevin Karplos. And the other route of this enterprise is really through Sean Eddy. So the way we do it, same way as before. Now going into the letters, we have an insertion deletion state or an alignment state. A match state is what it means. Then we get the initial probabilities by the observation of states that we have at that particular position, and then we build up this hidden Markov model. Sam is the one from the Hausler group, or Kevin. I believe I'm going to completely go over it in a very fast way and say that, oops. So you build up the profile. The way you assign these probabilities is you essentially learn the family step, and it's one step at a time. You learn the transition probabilities. Once you have the family, the transition probabilities, now you come with a new model, and that fits immediately. You can immediately say, what's the probability of fitting into that hidden Markov model? Okay? Where do you get your...
You mean new sequence or new model?
Your new sequence.
Okay.
Where do you get the family from? All the slides that I skipped explain in a little bit more detail how you actually go about evolving the transition probabilities and the priors and how you work this model out. But they do not explain how do we start. And we start with a family. Where do we get the family from? From the same place we get the cluster. That means from Psi-BLAST, most likely. So there are, in the olden days, so when Kevin or when David Hausler, when they started, they could still do them by dynamic programming by Smith-Waterman, but today this is not really feasible. So here's another. Have you ever heard of tea coffee? So hammer, you know what do you know hammer? Have you ever heard the word hammer? I mean, not that's not what I mean. The program. So what is it used for? Does anybody know? So hammer is clearly, Psi-BLAST everybody knows, Cluster all biologists know, and hammer, nobody knows the name, but almost everybody uses PFAM. So the PFAM is done with hammer. That's why there were these two competing suites of hidden Markov models for alignments, and this one survived simply because everybody uses PFAM. And that, in fact, also got better over time. And here is a completely different idea. Tea coffee, genetic algorithm. Any idea what that is, or how would you use genetic algorithm for? Okay, let's begin with the players. So the players in this one here is Cedric Notre Dame, Des Higgins. You have seen Des Higgins before. That's the cluster guy. So now you have and Jaap Heringa. Heringa was part of the initial group. So Cedric was the one who worked as a postdoc. He did his PhD thesis with Des Higgins. He in fact was the first one at the EBI, the European Bioinformatics Institute. And when so we are talking 90-something, 92. We are talking about a moment when already your supervisor wrote the best alignment program that the world uses at this point. So now then you're Cedric Notre Dame, so you have to prove your supervisor is wrong, or you can do something better, right? That's always what you have to do as a student. You have to change the world and begin with your shocking your supervisor by doing something that they don't understand. By the way, now Cedric is in Barcelona, and his institute this is his institute, and that is not a photomontage. So that is where you get from the institute. So the coffee is down here, and then that's the beach. Just beautiful. That's in fact, by the way, one of the reasons why Cedric ended up. He's a clever guy. Dummies for bioinformatics, by the way, Cedric Notre Dame. You begin with a library of local and global pairwise alignments. Mind you. So this is the point where everybody else starts with a family. And here we start with a bunch of alignments of all types, and in fact global and local. So we begin with something that everybody else throws away. We have two different types already there. We take this bunch of alignments here and put it into a grinder and get a multiple sequence alignment. So the way we do that is at every single position we have a progressive alignment strategy, so that is essentially the Doolittle idea implemented in Clustdal, where you have pairs and pairs and pairs and pairs and you go further. But now the difference is that as opposed to Cluster W, where every pair alignment sort of goes like this, you can now in a dynamic programming jump. You can take parts out from somewhere else and put them back and forth. In a genetic algorithm, you simply copy bits and pieces. Okay? And then these bits and pieces are taken from the input of different alignments. So you could take this pair alignment of the same pair, and swap things around. And then you have some criterion of some objective function by which you decide these you take, these you don't. And you, at every single step, you take some that maybe not entirely right, but that allow you to evolve into something new. And the ultimate output then of tea coffee is something that Clustdal could have never done. You move around the step and you essentially get the right alignment where Clustdal goes wrong. That's one beauty. The other beauty is that one major problem in hidden Markov model dynamic programming in PsyBlast is that we always assume what is the alignment at position I is independent of I plus 1 and I minus 1. So you align a protein position by position, residue by residue. And you the rest of the protein has no bearing on this alignment. We know that this assumption is wrong, but this entire field of alignment is based on that assumption. Because in the formalism of dynamic programming, we cannot do it otherwise. As double dynamic programming from Willie Taylor, but this is too complicated. Nobody in the world understands it. And this here is one of the few ways to get out of that dilemma. Because through the genetic algorithm, you can swap constraints in. You can say you accept that particular swap if and only if it fits to some constraint. The constraint could be that the cysteines bind. There's something else in there that has a cysteine bond. So if you swap this one, you have to swap the other one too. So you have good structural constraints, functional constraints, and you can easily, in this formalism of the genetic algorithm, can easily put that into the machine. That's the beauty. And no other method can do that in a generic way.